Welcome to Arca's Neet Biology, and today's topic is Human Genome Project. Now, what we are going to discuss about Human Genome Project. Okay, what is the meaning of it? What is the history, aims of it? Okay, how the mapping of human genome is being done, and what is the outcome from the project. Now, what exactly is Human Genome Project? Okay, now the genome is nothing but the DNA which is being present in the humans. Now this project intention is to know the entire information which is present in the DNA. Cracking out the codon sequence which is present in the DNA is the major target for this project. What is the use of knowing the genome means? Okay, here if there is any mutation in the genome, with what particular type of disease the individual is going to be affected, or what diseases are being caused due to variation in DNA can be easily identified once we know the sequence of the DNA. So here, this Human Genome Project is being done by the International Collaboration Research Program in the year 1990, and this was being completed by 2003. And here, what is the goal of this Human Genome Project is to understand and study all the 3 billion DNA subunits, that is the nucleotide sequences, and to identify all the human genes and making them accessible to the biological study. Whoever is interested in research can go on to the site NCBI, and they can check the information of human genome. Not only human genome, they have also cracked down many other organisms like E. coli, C. elegans, Arabidopsis. All these sequences were also cracked by this Human Genome Project, and that will be very beneficial to the current research study. And now coming to the history of this Human Genome Project, this was being first set up by the Department of Energy and National Health, that is the National Institute of Health. Okay, this being again, there is international collaboration by many other countries like Japan, okay, Germany. All of them, they have been joined their hands and started funding to this Human Genome Project. And what is the aim of this Human Genome Project is they had the several goals to achieve. And the first thing is I wanted to identify all the human genes. That is, they have thought that approximately it would be 25,000 to 30,000 genes, and they should be knowing all the information about the human genes. And they also want to determine the sequence, base sequence. Okay, the information of the DNA should be cracked down completely. So that was the another target. And the third one and the most important one is to store the information in the database. Okay, as I told you, this entire information is approximately 3 billion chemical bases. Okay, this sequence storing is the big task. For that, several technical development or the tools are also being used for data analysis as well as storage. And again, it should be available to the users who are into the research, as well as several other private sectors also should make use of this technology, and that should be easily available. So this is the biggest task for the Human Genome Project. The next one is to address the ethical, legal, and the social issues which are being raised against this Human Genome Project. Because there were people who used to believe that the genome which is present in the human will be of a high secret and that should not be cracked out. Okay, so that awareness to the public should be given, and how important the human genome is going to be in the future times, so should be educated among the individuals. And coming to the human genome size, okay, all the information which is present in the DNA is in the form of deoxyribonucleic acid, nothing but the nucleotide bases like adenine, guanine, cytosine, and thymine. All the information should be known. Okay, and here what information we know is there is total 23 pair of chromosomes, where 22 are autosomes and the last pair is the allosomes. And each chromosome consists of hundreds and thousands of genes, and their size is being varied. Okay, and they been expected that at that moment there may be approximately 30,000 genes might be present. Okay, and they also thought that out of that only 25% of DNA is the one which is consisting of genes, and rest of them is extragenic. Other than genes, some genome is also present in the DNA. Next, what is the strategy to come up to do this human genome project? Okay, before going into the sequencing, what method they have been started up with means that is the Sanger dideoxy method as well as the shotgun sequencing method. Okay, and here for this all the this restriction endonucleases, the cutting of fragments and the separation of these fragments, knowing of base sequences, everything is being carried. And in this protocol, they also make use of yeast artificial chromosome as well as a bacterial artificial chromosomes as a vectors to introduce the DNA fragments and cloning. And finally, the outcomes of this human genome project. So from this genome project, it is clear that in the genome there is approximately 3164.7 million nucleotide bases, where the information is in the form of adenine, guanine, cytosine, and thymine. They also said that the average gene consists of 3000 bases, but the size may be varying from one gene to another gene. The largest known human gene is dystrophin with approximately 2.4 million bases. So the total number of genes estimated is 30,000 approximately. And they also said that all the humans are approximately 99.9% they have the same type of nucleotides, and 50% of the genes function are still unclear, and that is still under the area of research. Less than 2% of genome code for protein, and repeated sequence, that is the junk DNA, is a 50% of the human genome, and this contribute to the formation of new genes or to the modify and reshuffle the existing genes. 80 rich gene regions are gene poor, whereas in the genes mostly they consist of guanine and cytosine rich regions. And chromosome 1 has most of the genes, that is 2968 genes are present, whereas the Y chromosome has the fewest, that is 231. Scientists also identified about 1.4 million locations there are SNPs which are being varied with a single nucleotide polymorphism. The same tandem repeat is being varied with the change in single nucleotide. And these findings of DNA sequences helps us to study the common diseases which are existing in humans like cardiovascular diseases, diabetes, arthritis, and cancer. And this is being identified and as identified by human variation maps, which is being generated by HGP. If you like the video, share and subscribe. Press the bell icon to receive further notifications. This is Pravalika signing off.